Hey everybody, this is Miss Hunt, and we're getting ready for Module 6. This is our first full week in October, and let's get started. Okay, we're going to start with Chapter 8 this week, and it's another doozy of a chapter. View presentation mode. Okay, this is your metabolism, and your metabolism is how things get energy. And basically, if you're not making energy or needing energy, then you're not alive or you're dead. So we have to think about how our body's going to take in energy and make energy. Now, of course, this goes to cellular respiration. We just talked about the mitochondria. So this is kind of a big thing of diffusion, bringing things in. But of course, we're going to get to more of a technical chemistry with all this. So all I can tell you are the big things here. Expect vocabulary everywhere. When I'm doing my notes, as I'm reading through my PowerPoint, here it is. I keep a list of things. So I'm like trying to make sense of stuff and trying to, write it down as I go. But you'll expect vocabulary everywhere. Nowhere is it going to say, hey, what is the difference between a catabolic and anabolic reaction? But they're going to be used in your vocabs for your questions. So when you're asking about how enzymes work, then you need to understand if it's spontaneous or if it's exergonic or endergonic or, all these other words that you're going through. So that's when it really matters. So definitely working on the basis of your vocabulary. Just like with most of biology terms, there are two that come in pairs. So when you memorize a set that helps you, exergonic and endergonic are going to go together. Anabolic, catabolic go together. So synthase and hydrolysis go together. So try to remember who goes together and keep that kind of going. You will be asked questions on ATP synthase and ATP hydrolysis and enzymes. So in the beginning, we're going to talk a lot about this stuff called free energy, and you're going to say, this doesn't make any sense. You have to know that in order to understand enzymes. So the basics of that are going to help. I've given you the Bozeman videos, and the worksheets that go with it are great. So listen to me run through these notes, but go through those Bozeman videos and watch them, hear them repeat some of the stuff that I'm going through right now. And I think it'll help you understand the topics and content a lot better. All right, so let's get started. The living cell is a miniature chemical factory where thousands of reactions are going to occur. I just mentioned cellular respiration extracts energy. So that's important to think about how we take in food and then we get energy through this. Remember, everybody does cellular respiration. Plants, animals, we all do cellular respiration. Only the plants do photosynthesis. So this is how we're all making our energy. Cells apply this energy to perform work. And some organisms even convert energy to light energy. So look at this. Isn't this beautiful? This is bioluminescence. This is an algae that makes its own light. When I went to Puerto Rico years ago, and I got to swim in a bioluminescent bay, and all of this algae started to glow when it got agitated. When you put your hand in the water, it started to light up as it got introduced to more oxygen and the oxygenation of it. That's why it's right there by the waves. As the waves crash on the shore, they are hitting oxygen and they glow. It's beautiful. Here's a firefly. And lightning bugs are going to use this to make their body glow. But your metabolism is just all of your chemical reactions happening in your body. You'll hear it locally as like, I have a good metabolism. I can eat what I want, or my metabolism's bad, and that's why I gain weight. That's not really the sciencey term used correctly, but it kind of is. Your metabolism is all the reactions in your body, and going by different species or even different body styles, your metabolism will change. It's fast as you're little, and it slows down as you get old. So it does evolve. The metabolism is an emergent property of life. So remember, this is a characteristic of living things. All living things require energy, and your metabolism is how you have energy. All right, a metabolic pathway is really important in this unit to look at metabolic pathways. They begin with a specific molecule and end with a product. Each step is catalyzed by specific enzymes. So here you have a starting molecule. We have an enzyme that works with it. And then that leads to the second reaction and then the third reaction. So you have different enzymes that are coming in to make that product. That product doesn't work unless all of the enzymes come together. A catabolic pathway releases energy. So I put this picture over here because catabolic pathways release energy by breaking down complexes. And if you've ever owned a cat, they're constantly knocking things over. So it makes me think catabolic breaking down molecules. Cellular respiration is the breakdown. So cellular respiration is a catabolic pathway because it's breaking down glucose whenever oxygen is present and turning it into energy. Do you remember what that molecule is? Remember ATP? I hope so. All right. Catabolic breaks, but anabolic builds. So that would be my way to remember. Anabolic relations or pathways are going to build. They consume energy to build complexes. So here they've given you NADPH and ATP are going to be created and to build these molecules. Bioenergetics is the study of energy flows through living organisms. All right, energy is the capacity to cause a change, and energy exists in various forms. So this part of the chapter, it hits to more like physical science terms, but it is going to relate it to your body and to biological pathways. So kinetic energy is energy in motion. Thermal energy is the movement of the atoms. Remember, things move really quickly. They have a lot of heat to them, and heat is a thermal energy. Potential energy is by location, and then chemical energy is by the chemical reactions. And energy can be converted from one form to another. They've given you this nice picture to understand potential is at the top. We have kinetic in the movement. Okay. All right, but we have to relate this to the thermodynamics and to the human body in the end. An isolated systems, such as that approximated by a liquid in a thermos, is unable to exchange energy or matter to its surroundings. In an open system, energy and matter can be transferred between the systems and its surrounding. Organisms are opening systems. So the sun gives energy to the plants gives energy to your herbivores give energy to your consumers, and it works its way up the food chain. First law of thermodynamics, energy can be transferred from or transformed, but it can't be created or destroyed. So you can be moved, but it can't be created or destroyed. The second law, going here again. Okay, here's a chemical energy, where he eats the food, he's running. And this is big, that energy is lost as heat. And we're going to talk about that again, that's really enthalpy, which is a word for me I have to try to work on, enthalpy, where we get energy lost as heat in our system, okay? All right, the second law of thermodynamics is while there's entropies there, every energy transfer and transformation increases the entropy of the universe. So every time you pass it down, it's at a lower quality. So you're getting less and less and less. And that entropy is the disorder in the universe and in the system. All right, so most of it's lost as heat. Can't be created or destroyed, but it can be transformed. And the second law is when it's transformed, it's at a lower quality. Okay, here we go. All right, living cells unavoidably convert organized forms of energy to heat, and more disorder from that energy occurs. Okay, So we have to know if something is spontaneous or not. And I'm going to give you a couple examples of how we can tell if a reaction is going to be spontaneous. They can happen quickly or slowly. That's not the point. The point is that they will happen. Okay. Processes that decrease entropy are non-spontaneous, and they will occur if only if energy is provided, like cellular respiration would be a non-spontaneous reaction. Organisms create ordered structures from less organized forms of energy and matter. Organisms also replace the order. Okay, I'm skipping over this one. The evolution of more complex organisms does not violate the second law of thermodynamics, and entropy or disorder may decrease in a particular system, such as an organism. Okay, here we get into some of our weird math. The free energy change of a reaction tells us whether or not the reaction occurs spontaneously. All right, biologists want to know which reaction occurs spontaneous and which ones will need energy. To do this, they determine the energy and entropy changes that occur in a chemical reaction. All right, we're going to use this symbol right here, and this is change. They call this the free energy change. They also call it the Gibbs free energy change. The guy who discovered it, his last name was Gibbs. I can remember this because Gibbs was the high school that I went to way long ago in a galaxy far, far away. So the Gibbs free energy change, also called just the free energy change, is written by the triangle and then the G. All right, a living system's free energy is the energy that can do work when the temperature and pressure are uniform, such as in a living cell. So free energy, I wish they would just call it energy. So this is just the energy that is available. All right, the differences between enthalpy and entropy. Enthalpy, we're going to call it this letter H, and it refers to the measure of total heat. So like that bear that was giving off the heat was the enthalpy. The entropy is denoted by an S and refers to the measure of level of disorder in the thermodynamics. So every time energy gets passed down our chain. All right, the change in free energy during a process is related to the change in enthalpy, which is the change in energy. So here you have a total change in your energy, there's your enthalpy, and change in entropy, there's your change in S, and the temperature in Kelvin with the unit T. So here's our formula. It may look crazy. We're going to do it in class on Wednesday and Thursday. Okay, so here's your change in free energy is negative for all spontaneous processes. So that's what the first thing I've written down in my little notes here, that this Gibbs free energy is negative for all spontaneous processes. For processes with zero or a positive, they are never spontaneous. So if it's negative, it's spontaneous. And if it's zero or positive, then they are not spontaneous. Spontaneous processes can be harnessed to perform work. So when you think about your body, do you want your body to do jobs that don't require any energy to get started? Well, of course you do. That makes it a lot easier for it to happen naturally. All right. Free energy is a measure of the system's instability. All right. I'm trying to think if there's anything on here I wanted to go over. During spontaneous change, free energy decreases and the stability of a system increases. Equilibrium is a state of maximum stability. Okay, so here you have a nice diagram. We're showing the gravitational motion, so this is like a physical change, diffusion, which would happen naturally, and then a chemical reaction that would happen naturally. Here's more free energy, so we have less stable and the greater ability to do work. The guys on top of the diving board here, we have atoms tightly packed together and the chemicals tightly packed together. In a spontaneous change, the free energy of the system decreases. The system becomes more stable. The release of energy can be harnessed to do work. Okay, less free energy is down here with a lower Gibbs, a G, and more stability. Okay, All right, and this goes through that same thing again. The concept of free energy can be applied to chemistries of life processes. An exergonic reaction proceeds with a net release of energy and is spontaneous. So I wrote this one down again too. Exergonic would mean that that it's spontaneous. So that means that it is negative, because spontaneous is we're negative. So our free energy is less than 0. In an endergonic reaction, it is non-spontaneous. So our change in free energy is greater than 0. So positive or not spontaneous. And endergonic requires energy. I think of exergonic is the release of energy. Endergonic is taking it in an absorption of energy. All right, this is something we're going to see later on too. So we're looking at the change in enthalpy, and it tells you whether it's an exothermic or endothermic. And we see the change in entropy, and then whether it's spontaneous or not. So you start to see a theme here. Something's exothermic or exergonic, it is going to be spontaneous. Okay. Yes, I'm checking my notes to make sure I'm saying everything right. All right, it will be spontaneous. So these will happen automatically. So here, like if I mix just salt and water and I leave it alone, it'll naturally spread out and diffuse in that container. And it is spontaneous. So here too, these reactions are also going to be spontaneous. The endergonic is not spontaneous, no, and these require energy. So taking glucose and turning it into starch is going to require energy to do this. Okay. All right, this looks similar to an enzyme reaction rate. So we have free energy on the side. So as we go up, we're increasing energy, and we go down, we're decreasing in energy. So here we have the reactants happening, energy is being added, and we have a product. All right, in an endergonic, you have to add energy. Okay, energy is coming in order for us to get the product. Okay, and again, the exergonic, we are talking about less than 0 because we want it to be a negative for energy to be released. And endergonic, it's greater, the Gibbs free energy is greater than 0. Okay, there's the same reactions again. Reactions in a closed system eventually reach equilibrium and can do no work. All right, this was another example of getting our heads around the physical stuff. So if I think about this as a dam of water, having a high potential of energy right here, and it turning a turbine and coming down. All right, this means that the Gibbs free energy is less than 0, and this would happen spontaneously. So we would have energy be released naturally. It doesn't require any work. Here, you can see we need to have some work. This is going to be equal to 0. So the first picture is less than 0, so it's spontaneous. This represents exergonic reaction. All right, cells are not in equilibrium. They are open systems experiencing a constant flow of materials. A defining feature in life is that metabolisms are never equal and never reach equilibrium. A catabolic, remember cat knocks over, they break and release energy. So here we have an anabolic that's going to build something, and catabolic that's breaking it apart. Energy is released, energy is required. Okay. In an open hydraulic system, hydroelectric system, it just continues going down from one to the next to the next. And this is what we want our body to do. Man, if I could just have this spontaneous amount of energy and then everything goes to the next system, to the next system, to the next system, no energy is being required, this is really easy to think about and easy for my body to do. Okay. I almost think about it relating to like the roots. We talked about how that water pressure and how the water was moving up through the trees. It's very similar to that. The water is naturally going to move into the roots, and then because they have a lot of the water, it will move to the next level of cells, and then the next level and the next level. And it'll pull up without using any energy. It's going to be moving in order for this plant to take in the water and have it diffuse through. All right, 8.3 talks about ATP powers cellular work by coupling the exergonic and endergonic, which makes sense. I want to have something that creates energy and then followed by something that needs energy. So everything passes from one to the next really well. A cell does three main kinds of work: chemical work, transport work, and mechanical work. Okay? To do work, cells manage energy resources by energy coupling. The use of exergonic processes to drive the endergonic processes. So here we have an anabolic of breaking, excuse me, the bonds in catabolic breaking, anabolic builds and coming back together. So breaking apart to make the ATP, and then building back when you have the ADP. Okay. All right. ATP is adenosine triphosphate. Little note, remember that this is a nucleic acid. It has the phosphate in it. It's made out of a lot of things. So you're going to see a sugar ring in here. You see the nitrogenous bases. They're all coming there. Remember, the monomer for a nucleic acid is a nucleotide. All right. So it is here in one of these phosphate groups. In general, when we break bonds, we release energy. So this molecule right here is going to be popping off a phosphate down here, and that is going to break a bond and release energy. All right, so here we have adenosine is our amino acid, and here's our sugar, our ribose, and then we have the phosphate groups, the 3-phosphate for adenosine triphosphate. All right, and then we're going to add water. Okay, so if we add water, this is called hydrolysis. You should remember that. Adding the water called hydrolysis, and what it does is this causes a phosphate to pop off. So now this phosphate is loose, and now we have energy. Okay, all right, there's a lot of 3D models of ATP that the book gives you. All right, the bond between the phosphate groups of ATP's tail can be broken by hydrolysis, adding water. Energy is released from ATP when the terminal phosphate is broken. This releases the energy and becomes a chemical change. All right, so you remember at the beginning of these notes, I said a lot of vocabulary, which we've covered a lot. And then the most important parts were ATP and enzymes. So now we're getting into the meat of the ATP stuff that we have to know. And we're getting into the enzymes. All right, here's the adenosine triphosphate. Again, hydrolysis, water comes in. My phosphate gets broken down. Any guess what the ADP stands for? It stands for adenosine diphosphate. So here's triphosphate with 3. And here we've broken apart, we have diphosphate by adding the water. And this is going to give us energy. This is called the hydrolysis of ATP. All right, it's okay, the ADP doesn't go anywhere. We actually keep it and we take in another phosphate group. So it all keeps going in this like nice circle together. The 3 types of cellular work, mechanical, transport, and chemical, are powered by the hydrolysis of ATP. In the cell, the energy from the exergonic reaction of ATP hydrolysis, can be used to drive the endergonic reaction. Okay, again, I've repeated this over. Remember, exergonic is spontaneous and has a free energy change of less than 0. Endergonic, not spontaneous, free energy change greater than 0. Overall, the coupled reactions are exergonic. Okay, ATP drives the endergonic reaction by, here's our big word, phosphorylation. Phosphorylation. Okay, All right, phosphorylation is transferring a phosphate group. So if I give you a phosphate, then you've been phosphorylated. Phosphorylation, okay? So you're transferring a phosphate group to some other group. The recipient model is now called a phosphorylated intermediate. That's a great word. Okay, so here's glutamic acid that comes in. It needs energy to get broken down. All right, we're going to give it some phosphate, and now it's been phosphorylated, an intermediate. We can still see ADP is still here. Okay. All right, then we get ammonia coming in. I think that's ammonia. I better look it up. NH3, I believe, is ammonia coming in. Okay. I'm going to Google it right now. Oh, I'm right. It's ammonia. Okay. Ammonia comes in. All right. And the glutamine is made. Instead of glutamic acid, we get glutamine coming in. We still have the ADP, but look, our phosphate is back off. So now we have an extra phosphate. What's going to happen? He's going to go back to be coupled to make ATP. So this process keeps using the phosphate back and forth to make this work. Okay, here's the same thing happening again, but with math, which is great. We're going to be calculating this kind of math on Wednesday and Thursday in class, 6 problems. I'm so excited. Okay, glutamic acid and ammonia. We just did this. And no, see, there's ammonia. Okay. All right. Transport and mechanical work in a cell are also powered by ATP hydrolysis. ATP hydrolysis leads to the change in a protein shape and the binding ability. All right, so here we have ATP coming into a protein. We just talked about the plasma membrane. So this could be any of our transport proteins coming through here, but it's going to change its shape to allow a solute to come through. So here is our phosphate that's popping out, okay? Here is another example of a vesicle coming to the cytoskeleton. This is probably going to do exocytosis and coming out. It's going to be a mechanical work. Okay, there's the transport protein. There is the protein in the vesicle. Okay, regeneration of ATP. ATP is a renewable resource that is regenerated by the addition of a phosphate. So again, ATP breaks off the phosphate, ADP comes back around, adds a phosphate. The energy to phosphorylate ADP comes from the catabolic reaction in the cell. ATP cycle is a revolving door through which energy passes during the transfer from catabolic to anabolic. Catabolic breaks, anabolic builds. Okay. Okay. And now the activity you're going to do Monday, Tuesday in class, we'll go over all of this stuff again. And I hope it'll help clarify for your work stuff. All right, 8.4, we're getting to our enzymes. Enzymes speed up the metabolic reaction. We talked about enzymes in general biology, or honors biology. So let's go through some other things. We have the word catalyst here is a chemical agent that speeds up a reaction. So the reactions are going to happen, they're going to be spontaneous. They're going to happen no matter what, but they're slow. So if we want to speed up the reaction, then we're going to add an enzyme. Enzymes are catalysts. Catalysts are enzymes. Catalysts speed up a reaction. In my brain, I remember Katniss Everdeen from the Hunger Games. They called her a catalyst. She got everybody all fired up to go against the Capitol. She started the reaction. Catalysts are going to help start a reaction. Remember, enzymes are proteins. So when you did your macromolecules, an enzyme is a protein. Normally, proteins end with ACE, ASE. So if you hear something that ends with an ACE, it's probably an enzyme. Sucrase is going to break down sucrose. A lot of our ACEs go together. So that's kind of nice. Like sucrase goes with sucrose. So sucrase is the enzyme that breaks the sucrose, which is oses. Remember, oses are sugars. Okay. And it's a breaking it down, so it's catabolic. Here we have sucrose coming in. I'm adding water. All right. And water is going to break it apart into glucose and fructose. But we need sucrase, the enzyme, to come in here and do this. Every chemical reaction between molecules involves bond breaking and bond forming. The initial energy needed to start a chemical reaction is called the free energy of activation, or activation energy. All right. They give a big capital E with a little a because it's difficult. I guess because it's energy, the energy to activate. It'd be nice if it was activation energy. Activation energy is often supplied in the form of thermal energy that reactant molecules absorb from their surroundings. Okay, again, this is that enzyme graph. Okay, here's the activation energy. So here are my reactants, whatever molecules I'm going to be putting together or breaking apart. Here's the transition state. So it's going to go up. So I need more energy, and then energy gets released. And I have my products down here, okay, or what is being made, okay? The Gibbs free energy is less than 0. So this is spontaneous, and it releases energy. So this is exergonic. That pulls those vocab words from earlier. Okay, how an enzyme works is it reduces the energy needed to make this product, okay? In a catalyst, enzymes or other catalysts speed up specific reactions by lowering the activation energy barrier. Enzymes do not affect the change in free energy. Instead, they hasten, good vocab word, I added speed it up, reactions that would occur eventually. It's going to happen anyways. This just speeds it up. Okay. All right. Here's the graph that you have to memorize. Here's the amount of free energy. You can just think about it as energy. The progress of the reaction. Normally, the reaction goes up, and we need a lot of energy to start this off. And then we make our product. If we use an enzyme, you see it reduces the activation energy. Okay, here's the EA. So without the enzyme, it requires a lot of energy to get started. And with it, it requires less energy to get this started. It will sometimes make it finish a little faster. It does this with less energy, okay? Remember, this whole process would happen anyways, but it speeds up the rate the reaction happens. The reactant that an enzyme attacks on is called the substrate. So here is my enzyme. Here is my substrate. So if I'm binding to, we were using sucrose earlier. So if sucrose is what I'm binding to, the substrate would be the sucrose. My enzyme would be the sucrase, okay? Whatever the ACE enzyme is that would bind to it. These have to bind together. They're specific. One enzyme for whatever substrate it's binding for. No, it doesn't cross over to somebody else's. The active site is where these two bond, and when they come together, they call it an enzyme substrate complex. So when these two come together, the reaction is catalyzed by each enzyme and is very specific. The active site is the region on the enzyme where the substrate binds. So let's go back. This is the active site right here. Induced fit of a substrate brings the chemical groups of an active site into position and enhances their ability to catalyze the reaction, to get the reaction started. All right, here's another example. Here's the active site where this substrate is going to bind into. It makes the enzyme substrate complex and then opens back up and releases whatever product is there. It's either going to be bringing two things together or it's busting something apart. The big thing is the enzyme can be used over and over. So it can just keep doing this all day long of taking in that substrate. Here's a great video to watch the enzyme binding to a substrate. I put that in there for you. All right, the catalyst is the enzyme's active site. In the enzymatic reaction, the substrate binds to the active site of the enzyme. Enzymes are extremely fast acting and emerge from the reaction in their original form, which means they're reusable. They did not get damaged, they did not get changed, they can be used again. Very small amounts of enzymes can have a huge metabolic effect because they're used repeatedly. They can be used over and over. Here we go. Here's two substrates coming in. They bind to the active site and are held together in the enzyme substrate complex. Doesn't it look like a little hug? It's like hugging them and then releasing them. All right. The active site lowers the activation energy and the substrates are converted. And then we have the product is being released. And you're like, okay, where is this happening? It's happening all over your cells. It's happening in the cytoplasm. It's happening in the mitochondria. It's happening all over your body in all the different cells. The active site can lower an activation energy barrier by orientating substrates correctly, moving them correctly, straining substrate bonds, providing a favorable microenvironment, and covalently bonding to the substrates. All right. The rate of the enzyme catalyzed reaction can be sped up by increasing the substrate concentration. So the more of the substrate coming in, this will speed up your reaction. The other way you can do is add more enzymes. All right. When all the enzyme molecules have their active sites engaged, the enzymes are saturated. If the enzyme is saturated, the enzyme rate can only increase by adding more enzymes. So, how do you speed up a reaction rate? 1, you add more substrate. 2, you add more enzymes. That's it. Effects of local conditions on the environment or on the enzyme. Okay, this is big. I put it in red. Okay, 2 things that you have to know affect enzymes is temperature and pH. You must know these things. All right. Enzymes don't work at different temperatures and they don't work at the wrong pH. So if water has a different temperature, like we've been talking in environmental science about global warming and about the oceans increasing in temperature and the temperature of land increasing. And some people are like, who cares? Well, some plants and some insects are very sensitive to temperatures and they are going to rely on the environment to keep their temperature. And if the temperature goes too high or too low, their enzymes won't work and they'll die. Being warm-blooded, we have a larger range to when we work, but this is also why we don't work if our bodies get up to 107 degrees. Our enzymes don't work. And likewise, if our body gets down to 80 degrees, we're not going to work. It won't make it. Okay? Each enzyme has the optimum temperature. This is also why, let me go back to the temperature. This is also why when fall happens, our plants don't work. Our deciduous trees give up the leaves. They quit. They can't do this. They don't have that large temperature range. They can't keep the leaves growing and working, and the plant has to hibernate. The effects of temperature and pH, the enzyme has an optimum temperature, an optimum pH. That's it. So here you have the optimum temperature for a human. You should know that we're 37 degrees Celsius. We always say 98.6 degrees Fahrenheit. Just memorize 37 degrees Celsius is our normal body temperature. So here's our temperature down here. Okay. So remember, this is the independent. This is the dependent. All right. The temperature rate right here, where we have the optimum amount of rate of reaction, is at our 37 degrees. Over here, this is a bacteria. It's a bacteria that's living in these thermal vents. And it likes it obviously much warmer, and that's when it can do its optimum range of doing its processes. Also, likewise with pH, here's pepsin in our stomach. Pepsin likes a pH of, it looks like 2 to work properly. You can see that on the pH scale, remember acids and then bases. So it looks like pepsin, I'm going to move me over here. Pepsin likes a very acidic environment in our stomach, but trypsin is over here, and trypsin's at a base of 8. So it likes a pH of 8. So it does not like it acidic, but it likes it more basic. And this is in the intestinal cells. So each of these enzymes are only going to work. So if I happen to get pepsin in my intestines, it wouldn't work. And if there would be no way to get intestinal enzymes in your stomach because of a sphincter that would stop it from coming in, but they wouldn't work. Okay. And again, here's the optimum temperatures for humans. For bacteria, okay. Cofactors are non-protein enzymes helpers. Okay, they work with them, cooperate, two coming together. Cofactors may be inorganic, such as metal in ionic form, or organic. Organic cofactor is called a coenzyme, and coenzymes can include vitamins. It's a lot. Okay. Hey, I'm recording. I'll talk to you in a minute. Oh, okay. Enzyme inhibitors. Competitive inhibitors bind to the active sites on an enzyme, competing with this substrate. Non-competitive inhibitors bind to other parts of the enzyme, causing the enzyme to change shape. I'm going to show you a picture in a minute. But this was kind of cool, kind of not. Pesticides work this way. So let's take a look at it. So here we have a normal substrate that's supposed to bind to this enzyme, but an inhibitor, a chemical, a protein, comes in and it stops the substrate from binding. It's a competitive inhibitor. You can also have a non-competitive inhibitor, and it basically binds to another spot on the enzyme and changes it so it doesn't bind. So this is what a pesticide does, is it stops the enzymes from working and then kills your insect. So it's just stopping slowly the enzymatic activities. The evolution of enzymes. Enzymes are proteins encoded by genes. Changes and mutations in genes lead to changes in amino acid composition of an enzyme. Altered amino acids, particularly at the active site, can result in new enzyme activity or altered substrate specificity. Under environmental conditions where new function is beneficial, natural selection would favor the mutated allele. For example, repeated mutations and selections on this enzyme right here, I'm not even going to say it, in an E. coli result in change in the sugar substrate under lab conditions. So what would be the benefit of changing or mutating an enzyme? With E. coli, if it's not a benefit to us, it's a benefit to the E. coli. If the E. coli gets changed, then anything that we have that would detect E. coli, or if we were to spray something to kill the E. coli, maybe we spray an inhibitor to go in and bind to the E. coli. If E. coli changes, then it's a different version and then nothing works and we have to start all over again. So not very cool if you're the E. coli, or sorry, if you're us and trying to fight E. coli. 2 changed amino acids were found near the active site. Okay, we're getting close. 8.5, regulation of enzyme activity helps control metabolism. Chemical chaos would result if a cell's metabolic pathway were not tightly regulated. A cell does this by switching on or off the genes that encode specific enzymes or by regulating the activity of enzymes. Allosteric regulations of an enzyme, allosteric regulations may either inhibit or stimulate the enzyme's activity. Allosteric regulations occur when a regulatory molecule binds to a protein at one site and affects the function of another site. So here you have like the inhibitor coming into an allosteric site. And this stops the substrate from coming in. And then we can have an activator come in that opens it up. Most allosterically regulated enzymes are made from polypeptide subunits, each with its own active site. The enzyme complex has active and inactive forms. The binding of an activator stabilizes the active form of the enzyme. The binding of an inhibitor stabilizes the inactive forms of an enzyme. So you could use this in medicine to stop something or to allow something to come in, to allow an enzyme to work or to stop an enzyme from doing its work. Okay. Okay. Cooperativity is allosteric because binding by a substrate to one active site affects the catalyst in different active sites. Okay. Kind of see where we are. Feedback inhibition. I've seen stuff about feedback and feedback loops a lot. In a feedback inhibition, the end product of the metabolic pathway shuts down the pathway. Feedback inhibition prevents a cell from wasting chemical resources by synthesizing more products than it's needed. So in a feedback loop, if a feedback loop is positive, it keeps moving forward. The feedback loop is negative, it's going to stop it. It returns it back to the way it was. So in this, a feedback loop being negative would be like an inhibition and it stops. Let me show you the picture. So here you have an enzyme, you have the substrate, but the product comes back and binds to the enzyme and stops any more from being made. So it binds to that inhibitor site and stops any more from being made. Localization of enzymes within the cell. We kind of talked about this earlier, but they are in different structures in order to bring order to the metabolic pathway. Some are in membranes and some are in like the mitochondria for the cell for respiration. So here they are. This part, we always draw the mitochondria as a bean with zigzags. We look at it a little bit different with an electron microscope. We see that these lines are actually called the matrix. And those are part of the enzymes that are going to be binded in here. Okay, these are the last few pictures. So what this is that the phosphate that's being bonded, and you can see that it's increasing and to a point until it stops increasing. It doesn't get any bigger over time. So it increases to a point and then it's going to slow down. Here's our enzymes. We did this graph over and over. And I guess it kind of related to this picture of the penguins. It was really hard to get up there, but then you're just going down the hill. Okay. So again, once we have our reactants, they would naturally just fall down and go down that hill, kind of like the penguins. All right, this has been Chapter 8, the metabolism.